All right, today we are going to talk about cancer. This is another kind of short day. This is an extension of mitosis. And so do need you to know exactly what cancer is. Cancer is a dysregulation in the cell cycle and the division of cells. So it's uncontrolled and unregulated cell division. So what what does healthy regulation look like? So you should recognize this picture right here from yesterday, this wheel that starts right here, right where you see my cursor. It starts at G1 and it goes this way clockwise. All right, so G1, S, G2, and then M. So remember, G1, there's some cell growth. S is where DNA is going to replicate. And then G2 is where proteins are going to get made. And then M is mitosis. So throughout that G1, S, G2, M, we have these things called checkpoints. And these checkpoints are where proteins will come in and they'll check, okay, are we good to go? Is everything made properly? Can we safely move on to the next phase of the cell cycle? So there's stop and go signals that regulate the cell cycle. Usually, if there's something wrong at a checkpoint, the cell will then undergo apoptosis. If nothing's wrong at the checkpoint, then the cell proceeds to the next phase. All right, so we have 3 checkpoints. G1 checkpoint is the most important. It's controlled by the size of the cell. If a cell passes the G1 checkpoint, that means it's committed to being a dividing cell. So if it passes the G1 checkpoint, it's like, all right, let's start getting ready because we're going to divide. So there are some cells that don't actually pass the G1 or don't actually go through the G1 checkpoint. And I'd mentioned those yesterday. Those are your brain, your neurons, your muscle cells, and your liver cells, actually. If there's a stop at the G1 checkpoint and it doesn't mean it's because something is wrong, the cell will enter into a non-dividing state called the G0 phase. And so your nerve, muscle cells, and liver cells are all what's called back to G0. So the G1 checkpoint basically is the checkpoint that decides, are you a dividing cell or not? Okay, are you a dividing cell or not? The G2 checkpoint, so let's look back at that picture. So if you pass the G1 checkpoint, that means it's committed to cell division, goes ahead, it replicates DNA, some proteins get made. Ooh, G2 is right before mitosis. So I would write that down. G2 is right before mitosis. The G2 checkpoint is going to make sure that DNA replication was completed correctly. All right? It's going to make sure that DNA was replicated correctly and that there are no mutations and that the cell is an appropriate size to go through mitosis. The M checkpoint is in the very middle of mitosis, and it's called the metaphase checkpoint. And it's also called the M spindle checkpoint. So basically what it's doing is it's checking to make sure the spindle was formed correctly and that the spindle is attached to the chromosome. So if you can picture in your mind's eye, because you did your stop motion video yesterday, metaphase, remember, chromosomes line up in the middle and the spindle is attached to them. So I need you to underline kinetochore. Kinetochore is the protein that is, it's the protein that basically attaches the spindle to the chromosome. So it's the protein that attaches the spindle to the chromosome. So you definitely want to write that down. I'm going to ask about that. Protein that attaches to the, all right? And then this slide right here is just showing you a really good visual of that. This is metaphase. This is the kinetochore, which is the protein that attaches the spindle to the chromosome. And the M checkpoint is going to make sure that the kinetochore is attached appropriately. And then what happens right after metaphase is anaphase. So then the sister chromatids will end up getting pulled apart in anaphase, and then the entire mitotic event will then proceed to completion. All right, so what are some other things? So these are internal, I'm going back. That annoys some people when I do that. But I want to go back because I want to look at the picture. These are internal regulatory mechanisms. What about external regulatory mechanisms? All right, and so some of our regulatory or external regulatory factors or mechanisms, whatever you want to call it, include anchorage dependence, density, and density dependence. And then you have 3 blanks to fill in. Don't fill in those blanks yet because that's not what they go with. I just want to show you from the picture, anchorage dependence means a cell will not divide unless it's anchored to something. So when you think about cancer cells, when they become like mobile, sorry, I couldn't think of a word for it. When they become mobile and they start traveling through the blood, they've lost anchorage dependence because dividing cells normally are not just like hanging out, like flowing through blood vessels. Density dependent inhibition means when it gets too crowded, the cells will then, a signal will be released or they'll stop dividing. All right, so those are 2 regulatory factors: is a cell has to be anchored to divide and a cell cannot be too crowded. So growth factors are an external regulation, proteins released by other cells that will stimulate them to divide. And those are, they're proteins. So released by other cells to stimulate them to divide. Density-dependent inhibition. Crowded cells will stop dividing. Surface cell proteins will bind to adjoining cells to inhibit growth. So some cells will release proteins that will bind to other cells and will inhibit their growth. And then anchorage dependence. Cells must be attached to another cell or to the ECM to divide. Remember what the ECM was? It was the network of proteins and carbohydrates on the outside of the cell. And it's what I said looked like the like barbed wire on the outside of a prison. So that's what the ECM, extracellular matrix. If you don't remember that, then go look up a picture of it. So basically what cancer is, it's going to be a disorder in which the cells are going to lose the ability to control growth because they're not responding to regulation. They're not responding to internal regulation, the checkpoints, and they're not responding to external regulation, anchorage dependence, and density dependence. It usually takes about 5 to 7 genetic changes or mutations for the cell to transform into being cancerous. So DNA mutations cause cancer. And so these cell cycle checkpoints can get messed up, but anchorage dependence and density dependence also gets messed up. The cells no longer respond to it. And so how does a DNA mutation cause a cell to ignore the signals on, hey, you're not anchored to anything, you can't divide, or hey, we're too crowded here, you can't divide? So a DNA mutation, DNA codes for proteins, and remember protein folding is very important. So if DNA is mutated, the protein ends up misfolded, and therefore the protein can't do its job. So if the protein is like, some proteins are like the police officers of the cell cycle, coming in, making sure that, the cell is stopping at the red light, all the checks are getting done, and that they can proceed when everything looks good. They're not dividing when things are too crowded, not dividing when they're not anchored to something. So basically the law enforcement of the cell cycle is no longer, and it no longer works. And so the protein that would stop the cell cycle is faulty and can't work. So this all has to do, cancer ultimately has to do with misfolded proteins because of genetic mutations. And so once again, the cells are, they lose anchorage dependence, density dependence, and then the checkpoints just aren't working anymore either. And it's several genetic changes that occur before that happens. All right, a tumor is going to be a mass of abnormal cells that are rapidly dividing. If the tumor is benign, the cells are going to remain at the original site. If the tumor is malignant, that means that it's going to invade and impair the function of 1+ organs. So the question comes up, if a woman if a woman is in her 50s or 60s or something like that, and she has breast cancer, like what's the big deal, right? She's not breastfeeding anymore, you know, they aren't like as functional anymore, and she has a tumor. When does breast cancer become really dangerous is when the tumor, the breasts are really close to the lymph nodes that are in the like the armpit area. And so that's the first place that breast cancer will invade are the lymph nodes. And once they invade the lymph nodes, well, now they have actually invaded like a transport system because we have kind of like your blood circulates, your lymph nodes and lymph, which is white blood cells, also circulate. So then those cancer cells are able to become mobile. Metastasis. You can hear it said when cancer metastasizes. I've heard people say it like that before too. That's when cells separate from the tumor and travel through the circulatory system, whether it's the lymph system or the actual blood circulatory system, travels to other parts of the body. And then this is showing you breast cancer and just how a tumor can grow from a single cell. It invades neighboring tissue and then it spreads through the lymph and then it gains access to the blood vessels and it can establish tumors in other parts of the body. All right, so just a little bit about breast cancer. It's the second most common form of cancer in the United States, and it's the most common form among women. In 2012, the results of the genomics approach to profiling types of breast cancer was done. So this is why they say if you have a history of breast cancer in your family, you can actually go get a genetic test to see if you have the BRCA gene. And the BRCA gene is a good indication that at some point you will have breast cancer. And I always tell this story because it's the only person I know of who's done this, but a lot of women do it. They'll get a genetic test, and if they test positive for this BRCA gene, they'll go ahead and they'll get a mastectomy. So they'll go ahead and have their breasts removed. Angelina Jolie, which, you know, the actress, she actually tested positive for the BRCA gene, so she went ahead and just got them removed and got some new ones made, I guess. For lack of a better way to say it, but and now she doesn't have to worry about it. So that's one of the good things about breast cancer is that it's the genomics have been profiled and put in a database so that if you get a genetic test, they can compare it to that to see if you actually have the DNA sequence that causes breast cancer. So that's pretty cool. And here's a picture of a scientist. I don't know why I put that picture in there because you just needed a picture. This lady's, you know, that's what she's doing. Anyway, so what are the factors that contribute to cancer? Individuals can inherit oncogenes or mutant alleles of tumor suppressor genes. So an oncogene, I would underline that. An oncogene is a gene that causes a protein, stimulates the cell cycle, and a tumor suppressor gene is going to suppress the cell cycle. So these are the proteins I was talking about, the law enforcement, the police officers of the cell cycle. Inherited mutations in the tumor suppressor gene, APC, is common in individuals with colorectal cancer, and then the BRCA gene that I was just talking about. So either one of these a person could get tested for to see if they have a strong predisposition. Right? DNA breakage can contribute to cancer, thus the risk of cancer can be lowered by minimizing exposure to agents that damage DNA. So ultraviolet radiation, chemicals found in cigarette smoke, and it is believed that viruses also play a role in about 15% of human cancers because what they do is they inject or they release genetic material into the cell, and their genetic material can get incorporated into the host genome and altering DNA sequences and therefore causing disruption in tumor suppressor genes or causing oncogenes to be there. Converting all I want you to know is that they can cause an oncogene, which is a gene that stimulates the cell cycle, and then they mess up the tumor suppressor genes, the ones that stop the cell cycle. So what you have here is a recipe for uncontrolled cell division. And so this leads me into our next topic, which is Henrietta Lacks. Henrietta Lacks Sometimes mistakenly called Henrietta Lakes, Helen Lane, or Helen Larson. I've never heard her mistakenly called those names before, but I think that that has actually happened. So she was an African American tobacco farmer born in 1920, and she died on October 4th of 1951 from cancer, and it was cervical cancer. Cervical cancer is what women get checked for. So when women are 18 or older, they go every year to the OBGYN and they get what's called a pap smear. And what they do is they scrape the cervix and they send the cells to a lab to see if they're cancerous. So obviously, if every woman is supposed to get a check for this every year, once they turn 18, is obviously something that's very common. Okay. And so what they did was without her knowing, they cultured her cells. So they took her cells and they created the first known human immortal cell line for medical research, and they're known as the HeLa cells. And here's a really cool picture of what they look like under a fluorescent microscope. So why are they considered immortal? It's because, remember, when cancer cells divide, and telomeres get cut. So remember, telomeres are the things like the aglets at the tips of chromosomes. They get cut, and that's supposed to happen because if they get cut a certain number of times, the cell undergoes apoptosis because it was too old. We don't want it to divide anymore. But telomerase in cancer cells is going to come back and add telomeres back. So the cell will never be triggered to undergo apoptosis, and therefore it will be immortal. So here's one of the labs that they culture her cells in. And so this is a really long text, but I'll paraphrase it for you. So at this point in time, when they cultured her cells, they had only been able to keep cells alive for a few days. And so it seemed like the scientists were spending more of their time trying to keep cells alive than performing actual research on the cells. But when they cultured her cells, they noticed that they behaved differently and that they didn't have to work very hard to keep them alive, and that they would multiply and then they would just never undergo apoptosis. They would never die. So what they do is they put her cells on a petri dish with, with like glucose and some protein, some amino acids and all of that stuff, like give them the nutrition that they need in the petri dish, and they'll divide and they live forever. And they still are in existence right now. And so with these immortal cells, they did a lot of research. One of the things that was discovered with the HeLa cells was the polio vaccine. You can write that down. The first human cells that were successfully cloned were HeLa cells in 1955. They were put in mass production, and they are being used around the globe for cancer, AIDS research, the effects of radiation and toxic substances on cells, gene mapping, in vitro fertilization, and fertilization is spelled wrong, sorry about that. And countless other, I would correct it, but it says fertilization, it's supposed to, and I'm acknowledging the typo. And countless other scientific pursuits. They've even been used to test human sensitivity to tape, glue, cosmetics, and many other products. 20 tons of our cells have been grown. 11,000 patents, and they've been used in 74,000 studies. A lot of money has been made from these cells. They've even been sent to space. So they wanted to see what the effect of space and the difference in gravity or no gravity had on HeLa cells and on actual cancer and on the activity of telomerase. If I can remember correctly, the outcome was telomerase activity declines when her cells go to space. But we can't really use space as cancer treatment, but it's still something interesting for them to have experimented with. So why was this controversial? Well, it was controversial because they never asked her if she was willing to donate these cells. And so in the early 1970s, her family, and this is a picture of her family, a lot of her family members, some of her family members, they started getting calls from researchers wanting their blood samples because they were desperate. They wanted to map their genetics so they can learn more about the heredity of the type of cancer that she had. And so you need her genetics, but you also need her family's genetics to find that out. And so her cells, Henrietta's cells, have been bought and sold by the billions, yet she remained virtually unknown while her family was too poor to afford health insurance. What? Her cells are being used to discover polio vaccine, AIDS research, cancer research. I mean, just tons of stuff. And her family doesn't even have health insurance. That's so messed up. So there's a book that's written. It documents the histories of both the HeLa cell line and the Lax family. It tells a riveting story of the collision between ethics, race, and medicine, of scientific discovery and faith healing, and of a daughter consumed with questions about the mother she never knew. It's a story inextricably connected to the dark history of experimentation on African Americans, the birth of bioethics, and the legal battles over whether we control the stuff we're made of. So very, very, very good book. And so, her, like I had said, the family was kept in the dark. When the existence of her cells was revealed, there's two articles, one in the Detroit Free Press and one in the Rolling Stones, just about how the confusion of her family members. How could her cells be? So this was a really big deal in the mid, 1975, 1976. This was coming out. The truth about it was coming out. And so right now, in her honor, there is a Henrietta Lacks Foundation that strives to provide financial assistance to needy individuals who have made important contributions to scientific research without their knowledge or consent. And so, permission to harvest a person's cells was not required, and that's how the cells legally could later be commercialized. They even took it a step further. In the 1980s, the family's medical records were published without their consent. In 1990, the court ruled that a person's discarded tissue and cells are not their property anymore and can be commercialized. In March 2013, German researchers published the DNA code or genome of a strain of HeLa cells without permission of the family. And so I think at that point they just kind of were fed up and had enough. But then they entered an agreement with the NIH, and they gave the family some control over the access of the cell's DNA code and a promise of acknowledgement in scientific papers. In addition, 2 family members will join a 6-member committee which will regulate access to the code. So I'm assuming that in order to access the code, a lab has to pay. So I'm assuming that they are getting some kind of financial gain from this. But I don't know, I still feel like, personally, I don't think they got enough. I just don't. So that is the story of Henrietta Lacks. It is a very interesting story, and if you ever want something to read, I would definitely read that book that I recommended. Let's talk a little bit about cancer prevention. Anyone can get cancer, but there's a way to minimize the risk. Don't smoke, legal or illegal. This includes hookah, chew, or secondhand smoke. Use sun protection like sunscreen. Exercise and keep your weight at an ideal level. Stress on the body can cause DNA damage, and being overweight can cause stress on the body. Eat 5 to 7 servings of fruit and veggies a day, and I'll tell people that, and they're like, whatever. I'll tell you why in just a minute. Always use preventative measures on, breast, testicles. You got to go into the doctor, guys, when you reach a certain age too, to check for prostate cancer. It's later than the women, but I think men, once they hit 50 or 55, have to start going in to get their prostate checked because of prostate cancer, and then getting moles checked is important. Practicing abstinence or using condoms. What's that about? That's about HPV and human papillomavirus. Human papillomavirus really is the main cause of cervical cancer. And getting vaccines, the HPV vaccine called Gardasil, that's something very important for people to get nowadays. That is something that, because I used to work in pharmacy, I remember when it first came out, I think it was like 10 years ago when Gardasil came out. So it's fairly new. It's actually fairly new, and it's a great thing. So HPV is a sexually transmitted infection, like I said. It will cause a person to be completely asymptomatic, so you don't know if you have it. There's 19 or 20 different strains. Some strains of HPV can cause genital warts, which I'm pretty sure if a person had that, if a male has HPV that causes genital warts, he will know. If a female has it, she probably won't know. And it just has to do with like the location and the visualizing of it, being able to see it. Some HPV strains can cause genital warts and cancer cells, and then some HPV strains can cause only cancer cells. So it's very important to get the vaccine for this and to practice abstinence and protection. So it can take, let's see, so the virus infects the cells, it takes several weeks for the infection to spread, and 10 to 30 years later can cause cancer. So it's kind of scary, and it's very silent. All right, let's talk about fruits and vegetables real quick. And I think that's actually the end. Yeah, almost the end. Yep, it's almost the end. So fruits and vegetables, this isn't like a trick to get people to eat them. This is literally like the truth. Fruits and vegetables have nutrients. It includes carotenoids, folate, vitamin C, vitamin E, selenium, flavonoids, and various other phytochemicals that can reduce cancer. So how do they do that? So fruit and vegetables contain a lot of fiber. And because they contain a lot of fiber, they can mop up. They literally act as a mop in the digestive tract to suck up and absorb harmful chemicals that could potentially damage DNA. And so if they're absorbing harmful chemicals, then it helps protect against DNA damage. Different minerals, remember we talked about cofactors and coenzymes. Those are found in fruits and vegetables. Well, enzymes are involved in repairing DNA. Enzymes need cofactors and coenzymes. So if you can think about it, fruits and vegetables are releasing minerals and vitamins in your body that help stabilize the active sites of the enzymes that are needed to help prevent and to repair damaged DNA. And then blocking of the formation of cancer-causing chemicals. I think that has to do with them acting as like a mop. And there you go. There's a website, in case you don't believe me, how eating healthy prevents cancer. All right. I hope you guys enjoyed that we did take a day to talk just about cancer and Henrietta Lacks. I do have a really great activity, HHMI activity for you to do on cancer. And then we will go ahead and call it a week. And you will have a test on Monday, and I will have sent out a review for that. So I will talk to you guys later.